For most of human history, agriculture changed at a pace that matched nature. Slow, incremental, and painfully limited by biology. Crops improved not because humans fully understood them, but because time eventually filtered out what failed. Farmers planted, observed, selected, and repeated the cycle across generations. Progress existed, but it was measured in decades, sometimes centuries. That system worked when the world was smaller. It worked when populations were lower, climates were more stable, and land was abundant. It worked when failure meant a bad season, not a global supply shock. But the rules of the game have changed, and agriculture is now operating under pressures it was never designed to handle. Global demand for food continues to rise, not linearly, but structurally. More people, higher caloric intake, more protein-heavy diets. At the same time, the resources required to produce that food are becoming scarcer. Arable land is finite, freshwater availability is declining, input costs are rising, climate volatility is no longer an exception, it is the baseline. Agriculture is being asked to do more with less, while absorbing risks it cannot control. For decades, the solution followed the same pattern. Scale harder, use more fertilizer, apply stronger chemicals, expand mechanization, push yields upward by brute force. This approach delivered results, especially during the 20th century. The original Green Revolution transformed global food production and prevented widespread famine. There is no denying its impact. But that model is reaching its limits. Yields are plateauing in key crops. Pests and pathogens adapt faster than chemical controls can keep up. Herbicide and fungicide resistance is spreading globally. Soil health is degrading under intensive practices. And extreme weather events are no longer rare anomalies, they are systemic disruptions. What once felt like optimization now looks like diminishing returns. At the center of this problem is biology itself. Crops are living systems, governed by genetic constraints. You can optimize irrigation, deploy AI-driven precision agriculture, and automate machinery, but none of that changes the fundamental genetic ceiling of a plant. If the crop cannot tolerate heat, resist disease, or efficiently use nutrients, the system breaks under stress. Genetics became the bottleneck. Traditional breeding tried to solve this, but it operates at the speed of inheritance. Crossing plants, selecting traits, stabilizing lines. Each cycle takes years. Each improvement drags along unintended characteristics. Breeding is powerful, but it is slow and imprecise by modern standards. As pressures mounted, genetic modification entered the conversation. GMOs promised a shortcut. Insert beneficial genes, accelerate progress, solve biological limitations directly. Technically, they worked. Economically, they scaled. But socially and politically, they ran into a wall. Public distrust grew. Regulatory frameworks hardened. The technology became associated with corporate control, opaque science, and unintended consequences. Whether those fears were justified or not became almost irrelevant. The perception stuck. GMOs turned into a cultural battleground instead of a purely agricultural tool. As a result, innovation slowed. While climate stress intensified and food systems became more fragile, genetic progress remained constrained by regulation, public resistance, and technical limitations. Agriculture kept pushing the same levers, even as those levers produced less impact each year. This created a quiet crisis. Food production did not collapse, but it became increasingly inefficient, fragile, and dependent on inputs that themselves were becoming unsustainable. The system was surviving, not adapting. And survival is not a long-term strategy. Here's the critical insight. The problem facing agriculture is not a lack of technology. It is a mismatch between the speed of environmental change and the speed of biological adaptation. Machines evolve faster than plants. Software iterates faster than genetics. Data moves instantly, while crops are still bound to seasonal cycles and inherited traits. Until that gap is closed, every other innovation hits a ceiling. This is why the next transformation in agriculture cannot come from the outside. It cannot be solved solely with better logistics, smarter sensors, or larger equipment. Those tools optimize the system around the plant, but they do not change the plant itself. To move forward, agriculture needs a way to accelerate biological adaptation without sacrificing control, safety, or public trust. That is the problem no one escapes. And solving it requires going deeper than fields, deeper than machines, deeper than supply chains. It requires going down to the level where the rules are written. At the level of DNA. For years, agriculture chased efficiency everywhere except where it mattered most. Machines became smarter, Sensors became cheaper, data became abundant, fields turned into dashboards filled with metrics, predictions, and optimization models. On the surface, it looked like progress at full speed, but beneath all that technology, the same crops were still growing. Precision agriculture optimized inputs, AI improved decision-making, automation reduced labor constraints. These tools reshaped operations, but they never touched the core limitation of the system. They improved how crops were managed, not what crops were capable of. That distinction matters more than it seems. A perfectly managed field cannot outperform the biological limits of the plant growing in it. You can deliver nutrients at the exact milligram, water at the perfect moment, and protect against pests with predictive models, but if the crop lacks genetic tolerance to heat, disease, or stress, failure becomes inevitable under pressure. As climate variability increased, this weakness became obvious. Heat waves pushed crops beyond their thermal thresholds. Droughts exposed poor water use efficiency. New disease strains emerged faster than chemical controls could adapt. Pathogens evolved, pests spread into new regions. Each season introduced variables that traditional farming systems were not built to absorb. The response was predictable: more chemicals, stronger formulations, higher application frequency, temporary fixes stacked on top of each other. This kept yields stable in the short term, but at a growing cost. Resistance increased, environmental pressure mounted, regulatory scrutiny tightened, margins compressed. The system was reacting, not evolving. At the center of this stagnation was genetics. Crop genomes define how plants respond to stress, how efficiently they convert inputs into biomass, and how resilient they are under unpredictable conditions. When those genetic instructions are misaligned with the environment, no amount of external optimization can fully compensate. Traditional breeding tried to bridge this gap. Breeding programs selected for yield, disease resistance, and stress tolerance. Over time, they delivered improvements, but they were constrained by two fundamental limits: speed and precision. Each breeding cycle takes years. Stabilizing traits across generations takes even longer. And selecting for one improvement often introduces trade-offs elsewhere. A plant bred for higher yield might become more susceptible to disease. Resistance to one pathogen could reduce performance under different conditions. Progress came with baggage. As environmental change accelerated, this slow, probabilistic approach struggled to keep up. The gap between what crops needed to become and what breeding could deliver kept widening. This is where genetic modification initially appeared as a solution. By inserting specific genes, scientists could bypass generations of breeding and directly introduce desired traits. From a technical standpoint, it was a breakthrough. From a systems standpoint, it solved real problems. Crops could resist pests, tolerate herbicides, and improve yields at scale. But the method created a new set of constraints. Gene insertion lacked precision. Genes were often added from unrelated species. Public understanding was limited. Regulatory processes became complex and slow. Trust eroded. The technology became politically charged. Instead of accelerating innovation, it narrowed the path forward. Genetics remained the bottleneck, but now it was trapped between biological limits and social resistance. Agriculture was stuck optimizing around an unchangeable core. This led to a dangerous illusion. Many believed that data, AI, and machinery would eventually compensate for genetic limitations. That smarter systems could outthink biology. That enough sensors and algorithms could override evolution. They cannot. Biology sets the upper bound. Everything else operates beneath it. If a crop cannot resist a pathogen, predictability does not save it. If a plant cannot tolerate heat, automation does not cool it. If genetics are misaligned with reality, the system fails gracefully at best and catastrophically at worst. Closing this gap requires a different approach. Not brute force breeding, not broad genetic insertion, not external optimization alone. Agriculture needs a way to edit biological instructions directly, safely, and precisely. A method fast enough to match environmental change, and controlled enough to earn trust. That requirement defines the next phase of agricultural innovation, and it sets the stage for a tool that does not work around genetics. It works on them. To understand why gene editing changes everything, one thing needs to be clear from the start. CRISPR is not an upgrade to old genetic modification. It is a fundamentally different tool, built on a different logic. Traditional GMOs worked by adding something new. A gene from another organism was inserted into a plant's genome, often with limited control over where it landed or how it interacted with existing genetic instructions. The result could be useful, but it was never elegant. It solved problems by addition. CRISPR solves problems by correction. Instead of inserting foreign DNA, CRISPR allows scientists to target a specific sequence within a plant's own genome and make a precise change. That change might disable a harmful gene, enhance an existing function, or tweak how a plant responds to stress. Nothing random, nothing external, just a direct edit. Think of it less like installing new software and more like fixing a corrupted line of code. The reason this works lies in a natural biological mechanism. CRISPR is based on a system originally found in bacteria, which use it as a defense against viruses. Scientists repurposed this system into a programmable tool. By designing a guide sequence, they can tell the molecular machinery exactly where to go in the genome. Once there, the DNA is cut. What happens next is the key. The cell repairs the break. During that repair process, scientists can influence the outcome. A gene can be knocked out. A small sequence can be altered. A regulatory switch can be adjusted. The change becomes part of the plant's permanent genetic instructions. No foreign genes are required. This level of precision is what separates CRISPR from everything that came before it. Edits are targeted, predictable, and testable. Researchers know exactly what changed and why. That clarity matters, not just scientifically, but economically and politically. Because precision changes trust. From a development standpoint, CRISPR collapses timelines. Traits that once took decades to stabilize through breeding can now be achieved in a fraction of the time. Instead of waiting for inheritance to randomly produce a desired outcome, scientists can go directly to the source. This does not eliminate breeding. It accelerates it. Gene-edited plants still go through field trials, performance testing, and selection. The difference is that the starting point is no longer guesswork. It is intention. That intention translates into real-world traits. Disease resistance can be improved by disabling pathways pathogens rely on. Drought tolerance can be enhanced by modifying how plants regulate water loss. Yield stability can be strengthened by optimizing stress response genes rather than pushing plants beyond their limits. These are not theoretical possibilities. They are measurable outcomes already being tested and deployed. Just as important is what CRISPR does not do. It does not create unnatural hybrids. It does not bypass biological rules. It works within the plant's existing genetic framework. In many cases, the resulting plant could have emerged through natural mutation or traditional breeding. CRISPR simply gets there faster and with far less collateral change. That distinction has massive implications. From a regulatory perspective, many countries treat gene-edited crops differently from older GMOs. If no foreign DNA is present, the plant is often classified as equivalent to conventionally bred varieties. This reduces approval time, lowers costs, and removes barriers that previously slowed genetic innovation to a crawl. From a public perception standpoint, the narrative shifts. Instead of genetic modification, the conversation becomes genetic editing. Instead of mixing species, it becomes refining traits. Instead of uncertainty, it becomes transparency. The technology becomes explainable, and explainable technologies are harder to fear. Still, CRISPR is not a magic button. Precision does not mean perfection. Edits must be validated, off-target effects must be monitored. Long-term performance must be proven in real environments, not just controlled labs. This is not a shortcut around responsibility. It is a tool that demands discipline. But for the first time, agriculture has a way to directly align genetics with reality. A way to respond to climate stress at the same speed it emerges. A way to adapt crops without waiting for decades. A way to evolve biology intentionally rather than hoping it catches up. CRISPR does not replace farmers, breeders, or agronomists. It gives them leverage, and with that leverage, the genetic bottleneck that defined modern agriculture finally begins to loosen. Gene editing stopped being a laboratory concept the moment it reached real fields. Once CRISPR moved from controlled environments into commercial-scale testing, the conversation shifted. This was no longer about what the technology could do in theory, but about what it was already doing in practice. One of the clearest examples is disease resistance. Fungal pathogens remain one of the most destructive forces in global agriculture. They spread quickly, evolve rapidly, and cause billions in losses every year. Traditional control relies heavily on fungicides, applied repeatedly across growing seasons. This approach is expensive, environmentally costly, and increasingly ineffective as resistance builds. CRISPR changes that dynamic at the genetic level. By editing genes that pathogens exploit to infect plants, researchers have developed wheat varieties with strong resistance to fungal blight. Instead of killing the fungus after infection, the plant becomes a poor host from the start. The result is fewer chemical applications, lower production costs, and more stable yields under disease pressure. The economic implications are immediate. Reduced input dependency improves margins. Lower chemical use reduces regulatory exposure. Yield stability increases predictability for producers, insurers, and supply chains. What looks like a biological tweak quickly turns into a systemic advantage. Another real deployment focuses on nutrition rather than protection. Gene-edited tomatoes have been developed to enhance nutrient profiles without altering taste, texture, or appearance. This matters because modern agriculture has historically optimized for yield and shelf life, often at the expense of nutritional density. CRISPR allows that trade-off to be revisited. More nutrients per unit of food means higher value without expanding land use. It also creates differentiation in commodity markets that traditionally compete only on volume and price. This is not just about consumer health. It is about economics. When crops deliver more functional value, pricing power shifts. Producers gain leverage, supply chains diversify, food becomes less of a pure commodity and more of a differentiated product. Regulation plays a critical role here. In several regions, gene-edited crops that do not contain foreign DNA face lighter regulatory frameworks than traditional GMOs. This accelerates deployment timelines and reduces development costs. Small and mid-sized breeding programs, previously locked out by regulatory expense, can now compete. Innovation decentralizes. Instead of a handful of corporations controlling genetic pipelines, more players gain access to advanced breeding tools. This reshapes the power structure of agricultural innovation, distributing influence across regions and institutions. However, this shift also introduces new challenges. As gene editing becomes more accessible, questions emerge around governance, traceability, and intellectual property. Who owns an edited trait? How are edits tracked across generations? How do markets differentiate between conventionally bred and gene-edited crops when outcomes look identical? These questions do not slow adoption, but they redefine it. Another consequence often overlooked is resilience at scale. Gene-edited crops are not just designed for peak performance under ideal conditions. Many are optimized for stability under stress. That stability reduces volatility across food systems. Fewer crop failures mean fewer price shocks. Fewer shocks mean less political and economic instability. In this way, gene editing quietly becomes a tool for risk management. Climate models predict increasing unpredictability. Gene editing offers a way to absorb that unpredictability rather than react to it. Crops can be tuned for regional stress profiles instead of global averages. This regional optimization matters. A drought-tolerant variety in one region does not need to match another region's genetic solution. CRISPR enables localized adaptation at a pace traditional breeding could never support. Agriculture becomes more modular, more responsive, and less dependent on one-size-fits-all genetics. Still, not everything is solved. Public acceptance remains uneven. Transparency varies by region. Long-term ecological effects require continued monitoring. CRISPR reduces uncertainty, but it does not eliminate responsibility. What it does eliminate is stagnation. The technology is already deployed. The impacts are already measurable. The consequences are already reshaping how food is produced, regulated, and valued. The only question left is not whether gene editing will spread, it is who will control it. Every major shift in agriculture has quietly reshaped power. The original Green Revolution changed which countries could feed themselves. Mechanization redefined labor and land ownership. Chemical agriculture concentrated influence in a handful of suppliers. Gene editing follows the same pattern, but at a deeper level. This time, the leverage point is genetics. Food systems do not fail first at the farm. They fail upstream, at the level of seed, trade access, and biological adaptability. Whoever controls those layers controls productivity, resilience, and long-term stability. CRISPR moves that control closer to the core than any previous technology. But unlike older genetic modification, gene editing does not automatically centralize power. Because the barriers to entry are lower, the outcome is not predetermined. CRISPR tools are cheaper, faster, and more accessible than traditional transgenic pipelines. Smaller breeding programs, public research institutions, and regional developers can now produce competitive varieties. Innovation no longer requires massive regulatory budgets or decades-long approval cycles. This creates two possible futures. In one, gene editing becomes another consolidation tool. Intellectual property tightens, trade ownership concentrates, access narrows, food systems become more efficient, but also more dependent on a small number of genetic gatekeepers. In the other, gene editing becomes a decentralizing force. Regions develop crops tuned to local climates. Public breeding regains relevance. Genetic diversity expands instead of shrinking. Resilience improves not through scale, but through variation. Which future emerges is not a technical decision. It is a policy choice. Regulation, transparency, and governance will determine whether CRISPR amplifies inequality or reduces it. The technology itself is neutral. The system built around it is not. There is also a geopolitical dimension. As climate stress intensifies, food security becomes a strategic asset. Countries that can rapidly adapt crops to new conditions reduce dependence on imports. Those that cannot become exposed to price volatility and supply disruption. Gene editing accelerates this divergence. Nations investing early in genetic infrastructure gain strategic insulation. This is not theoretical. It is already visible in how different regions classify and regulate gene-edited crops. Faster approvals translate into faster deployment. Faster deployment translates into competitive advantage. The private sector understands this. So do governments. Yet there is a deeper implication that rarely enters the public debate. Gene editing changes how society relates to biology itself. For most of history, humans adapted to crops. Then we shaped crops slowly. Now, for the first time, we can intentionally align plant genetics with future conditions rather than past environments. Agriculture becomes anticipatory instead of reactive. That shift carries responsibility. Short-term optimization without ecological awareness can still cause harm. Uniform genetics still carry systemic risk. Speed still requires restraint. CRISPR does not remove the need for stewardship. It increases it. The difference is that inaction is no longer neutral. Choosing not to adapt genetics in a rapidly changing climate is itself a decision with consequences. It locks systems into fragility. It amplifies risk. It delays solutions that already exist. The gene-edited green revolution is not loud. It does not arrive with dramatic announcements or visible machines. It spreads quietly, seed by seed, trait by trait. But its effects compound. Fields become more resilient. Supply chains become more stable. Food systems become less reactive and more intentional. Power shifts from those who scale inputs to those who optimize biology. The future of agriculture will not be decided by who farms the most land or owns the biggest machines. It will be decided by who understands, governs, and deploys genetics wisely. Because in the next era of food production, control does not start at the field. It starts at the code. And that code is already being rewritten.